Welcome back. Last time we spoke about the concept of effective length, represented by this symbol, which was equal to the length of a gene minus the mean fragment length plus 1. If anything about this equation seems unfamiliar or strange, please go back and watch the video on mean fragment length and effective length. In this video, we're going to talk about an application of effective length known as effective counts. This is one way to scale for the bias observed because of length. Scaling the observed counts the number of reads and fragments aligning to a particular segment of a genome to adjust for this length balance. And it has a very simple formula. It's the xi, the simple count, just the number of reads, the number of fragments aligning to a particular gene or region of the genome, times the length of that gene over the effective length of the gene. And I always find that it's better to look at these things with an example. So my example will include gene A and gene B. And I will say, firstly, you would never know this in an RNA-seq experiment, but let's just say that divine providence has told us there are 10 RNAs of gene A and that there are 10 RNAs of gene B. What we would know, though, is that gene A has a length of 600 base pairs and that gene B has a length of 1200 base pairs. We also know that the mean fragment length for this example is 300 base pairs. So since it's 300 base pairs, we can assume that the 600 base pair fragments of gene A, that these will split into 2 fragments per RNA on average, and that gene B will split up into 4 fragments per RNA. And so now we're able to calculate the simple counts. If we sequence it and we catch every single fragment, we're going to get approximately 20 fragments. So 20 reads will align to gene A and 40 reads will align to gene B. 10 times 4, 10 times 2. Makes sense. And with that, we can calculate the effective read. We can say that xi times the length of the gene over the effective length of the gene. We could also call this EFI. EFE. Very fun. All right, so if this, you can say 20 times 600 over 600 minus 300 plus 1. This represents the effective length of gene A. 600 over is approximately 300 is approximately 40. We do the same for gene B. 40 times 1200 over 1200 minus 300 plus 1 is approximately 53. All right, you might say, hold on there. We know that we have 10 RNAs, and 10 RNAs, those exist within a 1-to-1 ratio within the cell. Our effective counts don't look like they agree with the counts that we originally found. And you would be correct. But as I mentioned, we would never know that we have 10 RNAs of gene A and 10 RNAs of gene B. If we already knew that, we wouldn't have to do the RNA-seq experiment. And in fact, it is getting slightly better. It is correcting, in a sense, for this length bias. If you look, for example, at the normal counts, at just the standard how many reads align to this region, there's an approximately 100% difference between these two values. Whereas between 40 and 53, after we've done this scaling upwards, there's only approximately a 30% difference. So it is, in a sense, correcting for this bias. The other thing to consider is that effective counts are the simplest way, the simplest formula for correcting for this length bias. You often won't see people talking about or comparing effective counts within the literature, they'll use a more complicated way of correcting for length bias, such as FPKM. And we'll talk more about those in future videos. Now I've got a few more examples involving some murine genes. Those are genes that come from a mouse. It's also important to note that for this example, I am going to use a different mean fragment length distribution, or mean fragment length. This time it's going to be 400 base pairs. I'm also going to define these columns. So this second column is the length of these genes. This third column is the count. The fourth column, or the invisible fourth column, that will be the effective length. Then there'll be the ratio between the length of a gene and the effective length of its gene. And then finally, we'll calculate the effective counts. I'll catch you on the other side of all that calculating. We'll make a couple of observations then. All right, now we've gone through them all. Couple of observations. First, we notice that the scaling factor goes down as the length of the gene goes up. That's what we expected. That's what we saw in the very first example. So you can see that RGS20 has a smaller scaling factor than TCA1, for example. I should also note that I abbreviated some of the calculations here. If you want to see them out in full, go back to the first example in this video. We should also note, very importantly, that the length, the effective length, is 1 whenever the mean fragment length is greater than the length of the gene. Or even if this were, for example, 401, then you would still get an effective length of 1. Well, at 401, you would get an effective length of 2. At 400, you would get an effective length of 1. My apologies. So as you can see, it's actually very extreme in terms of its calculations, in terms of what the scaling factor as you sort of approach that mean fragment length. And at this, this number is probably a little bit unrealistic as to what is going on, this 4,004 counts of this gene. It is more to do with, the relationship between these two variables and the assumptions that this is making. The model works a lot better for genes that are a fair bit over the mean fragment length. And then I guess with this hypothetical gene, GM16041, just wanted to put an example in there. If the count is 0, then the effective count is also going to be 0. These two examples show that effective counts will often result in a decimal ending. This is okay, even though you would assume this is supposed to represent some kind of whole number count of RNA. Since we're already scaling, since we're already applying scaling factors, the decimal values are also a part of our final answer. Thank you for listening.